In this section, we will discuss cancer genetics. So, how are genetics involved in cancers? Well, one of the ways is through chromosomal rearrangements. An example of this classically covered on the boards is that of CML and what we call the Philadelphia chromosome. This is a balanced translocation in between chromosomes 9 and 22 that result in the production of an abnormal hybrid protein. This is a fusion protein like we talked about called the BCR-ABL oncogene. It stimulates leukocyte production and leads to chronic myelogenous leukemia. And here we see this is a good example of a translocation here where the ABL gene on chromosome 9 and the BCR gene on chromosome 22 get fused together and form this oncogene. Other cancer predispositions that we have: ataxia-telangiectasia, which can result in lymphomas as well as immunodeficiency. It's caused by the ATM gene, and it's a dysfunction in DNA repair. Bloom syndrome can lead to solid tumors as well as immunodeficiency, the BLM gene there, and it's the DNA helicase mutation. You'll note in all of these, or a good proportion of these, the problem is with DNA repair or DNA replication. Xerodermal pigmentosum, involved in skin cancer. XPB, XPD, XPA genes all involved there, and again DNA helicases and nucleotide excision repair are where the mutations are at. Fanconi's anemia shows a predilection for AML. You also see pancytopenia, that's where the anemia comes from, and skeletal anomalies such as a lack of radius. It's caused by mutations in the FACC and FACA genes, and again, these are involved in DNA repair. Other cancer predispositions, these are dominant familial retinoblastoma, causing retinoblastoma, and later on osteosarcoma, caused by the RB gene. This is the cell cycle regulation type of gene, so this is kind of a tumor suppressive gene mutation here. Li-Fraumeni is caused by defects in the P53 gene. You see sarcomas, breast tumors, brain tumors, and leukemia. Familial adenomatous polyposis, or FAP, colorectal cancer, the APC gene, fairly well known, fairly well characterized. Regulation of microtubule function is where the defect is at. Hereditary non-polyposis colorectal cancer, or HNPCC, also leads to colorectal cancer. Several different genes involved here: PMS gene, MSH gene, MLH gene, and this is the DNA mismatch repair defect. Again, problems with DNA repair. Familial breast cancer, breast ovarian cancer with the BRCA1 and BRCA2 genes, that's a defect in dsDNA break repair. And von Hippel-Lindau is known to be associated with renal clear cell cancer. It's the VHL gene, and this is involved in transcript elongation. Now, these predispositions for cancer come as part of what we call the two-hit hypothesis. In a normal person, we see up above, has two wild-type alleles for the TSG gene. And so in order for them to lead to tumor formation, they would have to have a mutation in one copy and then a second hit, a deletion or a mutation in the second copy, because again, these are recessive type of settings. You have to have two mutations because one would be sufficient. In this case, if this were to happen, you would see sporadic unilateral retinoblastoma. In the case of familial bilateral retinoblastoma, what you have is inherited a germline mutation from your mother or your father. So you're already starting with one hit. Therefore, all you need is one more hit instead of two hits, which makes you more predisposed to developing the cancer. Neurofibromatosis type 1 is another genetically associated disease. It's also known as von Recklinghausen's disease. Incidence is 1 in 3,000. 50% are actually new mutations, whereas the other 50% are inherited as an autosomal dominant type disease. The gene is found on chromosome 17, and it's known as NF1. It's a tumor suppressor gene. The mutation, there's multiple different mutations that occur in the population. Since 50% of the cases are spontaneous new mutations, that makes a lot of sense. It can occur anywhere along that gene. So what's the classic phenotype for neurofibromatosis type 1? Multiple cafe au lait spots, at least 6. Increased tumor susceptibility. You see axillary freckling, leash nodules in the iris, right? Surgical removal of worrisome skin nodules is the treatment. And here we see a picture of somebody that has von Recklinghausen's disease. Notice the multiple neurofibromatosis tumors. Multiple endocrine neoplasia, another syndrome that's involved with genes. These can be split up into MEN1 and MEN2A and MEN2B. For MEN1, also known as Wermer syndrome, you want to think about P's, all the P's. So look at that 1 as the long stem that would make a capital P. So you think pituitary, you think parathyroid, you think pancreas. Okay? Autosomal dominant mutation in the MEN1 gene, which really doesn't have any known function, but when it's mutated, you get MEN1 syndrome. Now MEN2A is also known as Sippel syndrome, and it's characterized by thyroid C cells. You can get medullary cancers, problems with the adrenal medulla leading to pheochromocytomas, and parathyroid malfunction as well. This is an autosomal dominant mutation in the RET gene, which is a receptor for glial-derived neurotrophic factor. MEN2B, similar to MEN2A in that there's problems in the thyroid C cells and in the adrenal medulla, but MEN2B is also associated with neurofibroma formations. This is another autosomal dominant disease, and again involves the RET gene. That concludes this section of the lecture. Next, we will discuss diseases of endocrinology and neurology. That concludes this section of the lecture. Next, we will discuss diseases of endocrinology and neurology. That concludes this section of the lecture. Next, we will discuss diseases of endocrinology and neurology. That concludes this section of the lecture. Next, we will discuss diseases of endocrinology and neurology. That concludes this section of the lecture. Next, we will discuss diseases of endocrinology and neurology. That concludes this section of the lecture. Next, we will discuss diseases of endocrinology and neurology. That concludes this section of the lecture. Next, we will discuss diseases of endocrinology and neurology.